Hello, everyone, and welcome to the final day of our science fair organized around big data applied to health. Today, we're going to start with a panel dedicated to drug discovery with new mathematical tools and database tools, drug repurposing, protein folding. We're going to talk about many topics, and we're lucky enough to have experts coming both from many fields, but also from academia and industry. So I'm very much looking forward for this conversation. And without further ado, I will now leave Kaspar Mossman, the moderator of this panel, take it from there. Thank you, everyone, and thank you for participating.
Good morning, good afternoon, bienvenue, wherever you may be. Hi, my name is Kaspar Mossman. I'm Communications and Marketing Director at QB3, which is a multi-campus University of California institute. QBI had its origins in the research division of QB3 at UCSF. And I'm here to facilitate the discussion today, and we're going to be investigating the big data approaches to drug discovery and repurposing in pharma. I've got this great panel of experts here. What I'd like you to do is briefly mention what challenges you're most interested in solving and what you do and where you do it, and then we'll start off with some topics. So Michelle, perhaps you could introduce yourself.
Sure, be happy to. I'm Michelle Arkin. I'm a professor of pharmaceutical chemistry at UCSF, and just upstairs from Kaspar's office. And we're really interested in our lab in a couple of things: in undruggable targets, so brand new target classes that people haven't been able to solve before for drug discovery, and also working with a group called the ATOM consortium. So ATOM stands for Accelerating Therapeutics for Opportunities in Medicine, and it's a big public-private consortium that's aimed at doing just this, using artificial intelligence and high-performance computing to really reimagine drug discovery as a rapid parallel activity. So starting from, I've picked a drug target, to I want to put that drug into man. Can we do that much faster using computation?
You're on mute there, Kaspar.
I'm muted. You know what? I'm just not going to mute myself here. Okay, great. Jim, go ahead.
So my name is Jim Bosley. I'm by background, I'm a chemical engineer, and I've been writing math models of disease physiology and pathophysiology and drug action for the past 20 years now. And I have a passion similar to what Michelle mentioned. I have a passion for increasing the efficiency of the drug development process. And so the models I use have helped to provide better decision support at each stage of drug development.
Great. Brice?
Yeah, hi. This is Brice Gaudillière. I'm an associate professor at Stanford University. My expertise is in systems immunology, and clinically, I'm an anesthesiologist. My lab uses various single-cell technology approaches, focused on proteomics approaches to understand how the human immune system responds and adapts to clinically relevant challenges and perturbations. And I can talk about several clinical scenarios later on. With respect to drug discovery, drug repurposing, we're analyzing really large single-cell data set, focusing on immune cells, which are really maybe the functional unit of how the human immune system functions. And with drug discovery, we're really going at the mechanisms of these drugs. How do they alter the signaling pathways and behavior of these cells to try to perhaps understand and identify molecular endophenotype that can narrow down and increase the efficiency of later on clinical trials.
Thank you. Julien?
Thank you, Kaspar, and good morning, good afternoon, everyone. I'm Julien Mamet. By way of background, PhD in molecular biology and pharmacology. I'm also an entrepreneur. I founded a company here in San Francisco called Adynxx that develops pain therapeutics and therapeutics for treating inflammation. And bio expertise, it goes from target discovery, target validation, all the way to mid-stage clinical trials. And I'm really interested lately in trying to use mathematical models or artificial intelligence to try to improve the way we can predict efficacy and safety of drugs in the clinic. And there is several components to that. And one of them is really trying to understand the difference between correlation as opposed to causality between biological relationship, and especially in networks. So I'm sure we'll talk about that.
Thank you, Klim.
Yeah, hi everyone. My name is Klim. I'm a QBI fellow here at UCSF, so I run a small lab, and my primary focus is I'm a structural biologist, so I'm really curious to understand how the molecular structure of proteins informs their function, and more specifically, I'm particularly interested in how the sort of metastable, flexible structure can inform function, and that's it's an area which is currently not really druggable, and it's not really even understood functionally what it does. And so I think figuring out that can lead to new candidates for drug targeting. And one of the major techniques I use is cryo-electron microscopy, which relies on highly intensive computation and image processing. And so therefore, I also have an interest in applying machine learning and artificial intelligence to actually process this data better, so that we can actually get at this sort of hidden states in protein structures and figure out how we could potentially maybe drug them.
Thank you, everybody. So we've got some questions that you all submitted originally to spur the discussion here. I think I'm going to start off with one of Michelle's questions. What are the biggest challenges using AI for drug discovery, and what are the short-term wins? And of course, you're going to have to define what AI means for your specific field, because it seems like there are many types of it, or many types of this kind of modeling. Michelle, would you like to respond?
Sure, thanks. Well, in the Adynxx consortium, we think about AI or machine learning or big data science in two different ways. One is modeling parameter, so something you can measure, a molecule binding to a protein, for example. And those we're pretty good at. I think the world is getting to be pretty good at that. The next frontier, so those are short-term wins. Can I make something that binds tightly to this target and not to this target? The long-term challenges are how to wrap that up into this kind of systems model that Jim was talking about, for instance, where we don't just say, does it hit this enzyme in the liver, but we say, is it toxic to the liver at this dose and time? So that systems model, trying to get all the pieces of the system together. That's the real holy grail.
Great. Jim, what are your, what type of AI are you talking about, and what are you looking for in the short term and possibly long term? You're muted, Jim.
Thank you. That's important to keep in mind. Let me maybe give you an overview of kind of my universe, and I think it dovetails with what Michelle just mentioned. A key economic element right now, I don't think there's a lot of super great correlation between some of like the big data and what are called AI and what I do. That said, there may be people listening that have already done what I'm saying. There may be people that are much more advanced, but I think that there's some opportunities. A key economic element in making drug development decisions is the quality of the decision support tool at each stage. And in early research, you don't have a very good decision support tool. You don't have any data about the target in humans. Yeah, you're depending on an in vitro readout or something. Does that correlate with the clinical outcome, which is the economic output at the end of the pot of gold at the end of the rainbow? We don't know. How do we do it now? Well, we might have an expert physician or a scientist make some estimation about whether the drug target's going to work. Is that going to be correlated with the clinical outcome? My experience is that there's a lot of expertise out there that people can say, no, that's not going to work, that's not going to work, and they're right. But to say something is going to work is a different matter, and I don't think we can do that. Over the past 20 years, we've begun to develop these big models based upon physics and physiology that incorporate a lot of different parts of the physiology. Two benefits: one, there's a lot of data for each portion of the physiology out there, so you can get the parameters Michelle mentioned. The other thing is, because the mathematics are based upon the physiology and physics, the model is more extrapolatable than a pure statistical model. You're deliberately creating these to get rid of that problem of a uncertainty gap. So now we've got these big models. I'll just cut to the chase and say, AI right now, I don't think is qualified to create these models. There's too high a probability of creating a bad model. Machine learning, natural language programming, great for coming up with all the data you need to get this thing. There's still some human curation. I think the real application of AI is to take the model you've created and identify weaknesses and give you ideas for how to fix it. As far as big data, I think that what you do is you take these models and again, you can filter the big data in a meaningful and efficient way. So you're shrinking the search space. So I'll shut up there and let somebody else talk for a while.
That's very informative. Thank you, Jim. Brice?
Yeah, great, thank you. And maybe the difficulties that I'm going to raise are partially related to what Jim just said. But we use machine learning in our lab in two different ways. In one way is for understanding biology, and for this, I think the algorithm that we're using are very appropriate, because we are in every sample that we are measuring, we're collecting millions and millions of data points. We're looking at single immune cells in blood samples. There are millions of them. Every single cell, we're looking at many parameters per cell. And so we have plenty of data points. Now that we use other techniques like imaging mass cytometry, we can do that in tissue. And we have tons of data to train these algorithms. So on a per sample basis, I think these AI types of approaches are really powerful to identify biological patterns, in a way that the human eye couldn't really do, but we can't see in thousands of dimensions. But now the issue is that what we really want to do is not just understand what happens in that one sample. We want to understand clinically relevant mechanisms. We want to identify features, specific features that are related to predictive, maybe, or related to a clinical outcome. Or when it comes to drug discovery, we understand what is the effect of that drug on that mechanism as it pertains to a certain clinical outcome. And now that's where the difficulty is, because we need many patients. We don't have millions of data points in one sample. We have, one certain type of data in one patient, coming from one patient. And now these techniques that we use are very expensive. It's not like you can generate millions of images, in millions of patients. We have a very limited number of training cohort and validation cohort. And I think the tools that we're using that are supervised learning tools are better and better, but there is always this compromise that we have to do between measuring a lot of things and having a certain number of patients that are accessible.
Thanks, Brice. Does anybody have any questions for Brice or Michelle or Jim at this point?
Yes, I have a question. I guess, in terms of applying machine learning or AI for all types of this sort of questions, how, what's a split between sort of black boxy type neural net things and, more like maybe what Jim was talking about, where you have explicit ODE type models where you actually know exactly how everything works and what the variables are. And, do we, in some of these cases, do we have enough high quality data to have a black box, sort of training? And do we trust it enough that we can start making decisions without fully understanding what the nitty-gritty, why is the algorithm gave you the particular hit or not, or do we not, and we just stay away from that and we only use explicit analytical model?
Yeah, so I'm an experimentalist, so I'm not really the right person to answer that algorithmically, but what I'd really like to know, we, like Brice, we take a lot of complex data, and it would be great to know which data we should pay attention to. And that active learning aspect of artificial intelligence that Jim was saying, that tell us where we're going wrong, tell us when our uncertainty is too high so that we can go back and collect more data to fill in that uncertainty gap. we'd like to do the same thing in the experimental side and have the machine tell us what data we shouldn't bother taking because it's not useful. How close are we to doing that? I don't know.
Yeah, I think you're bringing really the point that hurts, right? We would all want to use similar neural network type of approach, and let the algorithm tell us where should we look, that and what is really clinically relevant. The problem is that the data that often, maybe not in your lab, I'll tell you, in my lab, the data that we're entering are very imperfect. And there's a lot of variability in the data. And, if things are in the -80 for a few years, well, guess what? It just changes, and you don't have exactly the same sample that you're entering. So then you're using algorithms that are really very powerful when looking at gazillions of data points, but you're only giving them, a short amount and a small amount with a lot of variability. And I think there's a place to maybe try to cut down on that variability by introducing in these models prior knowledge, and I think many groups are using this from the fact that we are looking at very often mechanisms that have been studied for many years. There's a lot of understanding. It's not completely a black box. There's been a lot of hypotheses that have been derived before for the last 30 or so years of modern biology, and introducing some of that prior knowledge in these algorithms helps, I think, reduce the that variability. There's a danger to that too, obviously.
That illustrates, those are really good applications, I think, of trying to use a focused use rather than trying to solve the whole universe, focusing on a problem, and it can deliver a lot of value. I'm a little bit prejudiced against black box or neural net models for engineering purposes or engineering reasons. One is in systems with feedback, and that's one of the difficulties in biology. Systems with feedback, doing parameter estimation is extremely hard, because the feedback correlates the inputs with the outputs, and you don't know if it's the tail or the head. The other thing is if you think about models, what we're really not looking for is giving it inputs and having it tell us what the outputs are. What we really want to do is we want to tell it the output. We want a cured patient. Tell us what the inputs are. Tell us what the therapies are.
Yeah.
And that's called a model inversion, and that's what I did my PhD on. It's called nonlinear model predictive control, where you take a model and you essentially mathematically invert it to give you the inputs to run a physical system. In my case, it was a moonshine still, but the bottom line though is if you have under certain conditions, certain neural net models can be unstable in inversion, and you don't know. So I'm a little prejudiced against black box /neural nets. Not saying people haven't done wonderful things with them, and if you have, I'd be interested in learning. But there's some technical reasons why you want to be leery.
Thanks. Very interesting points. Knowing nothing about this field, I have a similar question, just the black box nature of it of the models, and I was thinking that's what we're talking about. What I'm thinking about is the neural net models and how they might give you the answer, but they don't give you the sort of physical understanding, and that's what one aspect that would be helpful. So I just like to keep getting everybody's perspective on how they would define AI in their particular area and what their short-term wins would be. Julien, would you like to tell us?
Yeah, of course. And I think we're all looking at the end of the day, trying to answer the same questions with multiple angles, it seems. And at the end of the day, it's when you want to develop a new drug to tackle, a biological problem, which target to use so it's functionally dominant, right? And there's this particular aspect of one target in a complex network. How are we going to find a way to know or predict better than what we're doing right now whether or not it's going to have a clinical input, right? And second to that, there's going to be the viability of this. What Brice was mentioning, you're going to get a lot of viability in human, much more than what we do in animals, because most of the time, we use inbred animals. So there is this component. So to do that, I feel like in mathematical models, AI have helped us to, at this point, help us start integrate complexity of biology. Let's put it this way. But I think, just like the other member of the panel, we're not there yet into understanding we're putting together this complexity, but trying to understand how it works, how it's get into life. That's kind of the problem. You can have pretty powerful algorithm, as people were saying, but what do they mean? I mean, they're going to produce networks and produce outcome, but what does this outcome mean in terms of drug development? So to me, that's really the challenge, trying to understand, once you have an output of this algorithm, what does it really mean from a drug development standpoint? If that makes sense.
Thank you. Klim?
Right. So yeah, again, so my dabbling in with AI and machine learning is a little bit on the other side, but I think it's fundamentally all really the same thing. So again, I do cryo-EM, and so that involves to obtain this molecular views of how, for example, a drug binds to the protein and how exactly it inhibits it, or to see where recently we had this work on the COVID where this viral protein was inhibiting one of the mitochondrial proteins. So we now can use that picture to design better drugs. But what people don't realize that to get to that picture, processing cryo-EM data, it involves thousands of grayscale images, which to untrained humans very hard to interpret, from which you end up with millions and millions of tiny images, which need to be clustered and denoised and pre-filtered and things like that. And so that's where my exposure to a lot of the machine learning has been. And my goal would be, again, to have this clustering algorithms be better, and denoising algorithms, for example. And similar questions to as we're discussing here is it became clear at some point that when neural networks just came out, you kind of wanted to just throw your images at it, and it would sort everything out. And theoretically, given infinite time and infinite compute, you can probably do that. But it's so much more efficient to actually pre-filter your data based on the prior knowledge and prior models, which we already know, so that you do what you know to the max. And then at that point, you feed everything into maybe even a black box to cluster proteins and conformations by different into different sort of structural states. Another very interesting thing, which again is plotting this data and conformational states on different manifolds and connecting them to give sort of continuous transitions between distinct states. That is also of great interest, and I think that's something that is, again, would be something as an early win, in the next 5 years or so, if we could do that better.
Great, thank you. What is evident is that there's many different types of expertise that are needed, many different perspectives on big data in pharma discovery and repurposing. And I mean, just look at the panel, everybody's got a different angle on this. So does anybody have any insight into what sort of skills they don't have that they're interested in working with? Or maybe you're working with somebody who complements your capabilities, and that would sort of help highlight what you need to bring to bear on the problem. So I guess I'll ask Michelle, what skills or technologies, this is originating from one of your questions, do you need to solve the problems that you face after the easy wins? And who might you be working with already on this?
Yeah, well, there are a couple ways to answer it, because I'm listening to the conversation, and I'm also interested in protein-protein interactions and how to modulate them. And the basic concept is that you have this whole network. This protein interacts with these 10, and then those 10 proteins interact with another 10, and you have these very complex networks. How do you know where to push on the system, which edge to remove or which protein to remove or to add to make that whole network bend in the right way for the disease? And so that's something I'm always talking to people and trying to start collaborations around how to address that question, because biology is extraordinarily complicated. I think what Brice is saying, millions of data points on thousands of cells, it's extremely complicated. And so, yeah, the black box in a way is kind of comforting. If it tells me the answer, at least I move forward.
But then you're wasting, like you have this vast computational power that we have now and a vast space to apply it to, and you want to make sure that you're aiming it at the right spot.
Yeah, that's right, exactly. Because drug discovery is really hard. If you think about it, like a drug molecule is 40 heavy atoms. It's a very tiny, simple thing, and everything that's going to happen to that drug is encoded in the structure of that molecule. So every piece of that molecule is very important. It has to bind to its disease target. It has to not bind to a toxicity target. It has to not be seen as food or poison. So many things that have to happen to it. And so when you couple that simplicity or that elegance with the complexity of biology, I think that is a great place for multi-parameter optimization, right? For this really data-intensive approach.
Okay, so who are you collaborating with now, or who might you want to collaborate with to match the small with the large?
So in the consortium, in the ATOM consortium, we have 5 national labs: Frederick National Lab, which is dedicated to cancer research, and then 4 of the DOE national labs that have really extraordinary computing power. And one of the reasons that they work in drug discovery, there are many good reasons, biodefense and things like that, but also it's one of the most difficult problems. And so it really pushes their computer science and their high performance computing. These are the 2 fastest computers in the world being trained on this kind of activity. So, so that's who we're working with. And it's really extraordinary, just different perspective.
Yeah, you had mentioned the ATOM consortium, so maybe we can return to that in a bit, and you can tell us a bit more about it. Jim, what sort of skills or technologies do you see as necessary that you might not have at the moment?
Well, so first of all, I'm an engineer, and engineers typically get very poor statistical training, but that's not just engineers, unfortunately. I think that there's some. UCSF has a phenomenally good pharmacometrics program. There's isolated islands of excellence in this area, but I think we need to up our statistics game. But then we have to realize that the kind of limitations of statistics in predicting outcomes outside of the sampled population. That's a big deal. So statistics, I think chemical engineering is a pretty good solid foundation for the ODE approach. The genome scale and metabolic modeling approach came up that way, but now we're getting good collaboration with very smart biologists in terms of protein-protein interaction, as Michelle mentioned, and transcription regulatory networks. So a biologist, an engineer, good statistician, and a whale of a big computer, and you can do some cool things.
Great. And don't forget your chemist.
Well, yeah, Michelle and I have Clement on the thing. Yeah, no. So I'm looking at the big picture. All I'm trying to do is exactly what Clement said: is filter things down to give you guys a soluble problem that you're going to be successful at. So if that's my job is to make you successful.
Brice, any thoughts? Oh, you're muted, Brice. Sorry.
I still think I'm using the Zoom platform. No, yes. So that's I have a few thoughts on that. First, like going back on what Michelle was saying about protein network, and the type of skills and statistical tools that are necessary for this, I think is really pertinent to the work that we're doing. We're looking at cellular networks and immune signaling networks. So really understanding, if a certain cell type activates or mobilizes certain signaling pathways, what is the repercussion of this in a different signaling pathway in a different cell types that are in the same environment and milieu, and how do these interact with each other? Because in the end, when you're using a drug, even if the drug is very specific, it may interact with that, one molecule. But guess what? The effect on that molecule will have a different effect on the cell, and that cell will talk to another cell, and it may not be exactly what you expect. We just had a study, a very simple study in humans on the effect of glucocorticoids on the human response to traumatic injury. And when you start looking, you wish you hadn't been looking, because it's, there's things that come up that you're like, well, maybe it's a good idea, maybe it's not a good idea. But all this points to the fact that this work needs to be transdisciplinary. We're one of the most exciting work that we do is through the Prematurity Research Center at Stanford, where when I thought things were complicated looking at single cell data, we got involved with Mike Snyder's lab, Steve Quake's lab, and Rob Tushani, Trevor Hasty, to really then start looking at how immune cells are interacting with all the other protein metabolites and cell-free RNA in the environment. And so now usually the complexity is even greater, and you're just begging for someone maybe upstream of all this to tell you a little bit, well, can you narrow down from, many millions of hypotheses to maybe a few hundred? And now you're, now you can, have the models that are going to be powerful enough to find the right thing. And then I'll finish with one last collaboration that I hope other people can, resonate with what other people are doing in the panel. But I just started working with Marina Sirota at UCSF, who was trained with Atul Butte, on using the data that they are generating from their in silico pipeline for drug repurposing, where they're really looking at publicly available RNA-seq data, coming down and narrowing down to mechanisms of existing drugs that are FDA approved, and relating this with a potential clinical outcome, and then having all sorts of new targets, new drug targets. And then the issue is that you have these beautiful in silico data, but you don't really know whether the mechanisms are going to be valid. You'll be able to validate that in an actual experimental assay, in a clinical assay. And this type of relationship is really important and really fruitful, I think, to kind of, again, narrow down the scope of the data.
Something that has occurred to me, which might come across as very naive, is what you've got these cells, which are essentially mechanical computers, and then you're trying to build these in silico models, which are silicon computers. And I don't know, it just, you've got the computer right there already. You can, like, if you could only make it make sense, but then you've got to know what questions to ask. Mm-hmm. Does that resonate? I mean, you've got the things already built. It's just that there's so much variability in the cell population.
Yeah.
Anyway.
Yeah. Very good point.
Okay, that's just me blurting in with my enthusiastic ignorance. Julien, what would you see as opportunities and skills that you would love to have or are needed in what you're doing?
So maybe, not different from what just been said in terms of type of expertise, but maybe a comment on how to operate, which is, the success of that field, this intersection of biology and mathematics and AI, is really trying to make two different worlds with completely different expertise and languages meet and talk to each other. And I think it's absolutely key that each party understand the other one. So, for example, I have a training in biology, not in mathematics. So what I am given, like a network, so gene network, and I'm putting on a software and I can visualize it. What does this mean? Because if I take one algorithm to look at it, it's going to have one shape. If I take another algorithm to visualize it, it's going to have another shape. How do you interpret that? So it's really about making sure each party understands what happened at every step of the processing, what data are being fed to the algorithm, what QC has been done, what have you been removing, what viability you have in your outcome. If you do some reverse engineering of networks, you can run, just run some data, you'll get one outcome. Run the same data with the same algorithm, you get some viability on the outcome because it's statistical. So how do you account for that biologically? How do you put that into your therapeutic strategy? So I think it's really about making sure you're really scrappy and trying to understand like every step of the way what's happening, basically.
Great. Klim, any thoughts?
Yeah, I mean, it's yeah, I mean, I think each of these fields is now so specialized, unfortunately, that it should really, you know, I'm not sure I can imagine a single person who is so trained in all of these fields to do all of it really well. And so I think if there's one thing which I would encourage people to do, or the skill that is needed, is this absence of fear of jumping the chasm and going to the other side. Where, and to give you an example, again, in biology, working at UCSF, I would argue there's a big need for hardcore computational scientists, for example. And that's why I started doing, this hackathon, where the idea is that can we just attract some people, we had people come in who work at companies where they take the satellite data imaging and then they basically automatically process it to track barges or whatever. And so then we had them come in and work with highly trained specialized biologists to track small molecules walking in the cells automatically. And it's the same algorithm. It just applied to a different thing. Very important is to work in this group so that no person does a mistake because they don't know that field. But bringing these groups together, I think, is really the way forward. And then an interesting thing is, once we started having these hackathons, it became clear after the second one that we should really bring a third group in, would be mathematicians who actually would develop deeper, sort of more abstract models, which then the computer scientists could, apply to this imaging data which biologists apply. And so I think, the training is that, we should embrace the interdisciplinary problem. I'm not sure, how possible it is to be, you need to understand the faults and the limitations of everything, but it's hard to be an expert in every one of these fields. And so I think it has to be really people just coming into these teams and working together and being really encouraged rather than intimidated by the complete unknown, which might be not your field.
It's almost like you have the orchestra that needs a conductor, right? But there is no conductor. So, I'd like to open it up to, you've all just raised such interesting topics, and I think you're best qualified to ask each other the questions at this point. Does anybody have any questions for somebody else here, or open questions that you'd like to hear responses on?
Let me jump in and ask that, because Michelle, kind of fiercely defending her expertise, talked about designing molecules and so forth, and Clement does, I think, some similar work. What do you guys need in order to zero in to make your problem from solving the universe, which is intractable, to solving something that's focused? What are the constraints that help you? Or is that too hard to say?
Yeah. In a way, I think it's like I went to this symposium a couple of years ago called Reverse Translation, Bedside to Bench. So for people who don't know, in translational science, we talk a lot about bench to bedside. So what we do in the lab making sense in the clinic. And this was backwards. It was a bit like what I think Brice was talking about with clinical data. So you start with the clinical data and you pick the targets from that. So what's hard for us experimentally is that our models are always simplified from the patient, and we're not 100% sure we're simplifying it in the right way, because if we simplify it in the wrong way, it will be irrelevant. So since we can't capture that complexity and we don't have enough patients and all the things that Brice was talking about, it's a really underdetermined experiment. We can't get all the variables that we need. So people helping us say, these are the right targets. This assay is going to capture the important pieces of that target. It would be very helpful in lack of failure in drug. So drug discovery, a lot of times, 50% of the time, it fails in clinical trials because we picked the wrong target. It just has no activity. So reducing that.
Anyone else? Brice, yeah.
have problem with technologies in general. Today is nothing new. Follow-up question to Michelle. What are the type of approach that, or do you use certain approach to kind of model, unknown patient population or unknown functional data that you don't have that, but you're using available data to model what you may have and increase maybe the data set and using a simulation type of method. Do you use any of this to kind of like virtually increase your sample size or
yeah, we don't. That's not really, I don't know about that. Seems like a good idea. Yeah, I don't have a good answer. I'm thinking about toxicity and safety modeling, but and what they do there is it's really physics. It's a series of flows in and out, flows of information, and trying to model, say, everything that happens in the liver. But yeah, I don't have a good answer for you. It's the big question.
Yeah, wouldn't it be nice if we could just generate, enough data to do all, a lot of trial in silico first, in other fields has been done, but we're very closely with Nima Gaipur, and he's trying to develop this and kind of generate out of the data that we have produced, generate new data, from patients that would look like the patient population that we have, in order to kind of like test more, basically increase the dimension of or the size of the data set. The problem that I see with that is that if you start with a lot of variability and a lot of basically what is a flawed initial training set, then you're only increasing the data set with the same type of flaw. If you don't have enough diversity, if you don't represent the entire patient population, you're always going to be, driven by that initial data set. And I'm just trying to always, ask other people if they use this kind of approach to see if they figure it out.
Brice, let me just, I want to address that, because that's kind of why I like the QSP approach, because you're using the whole universe of data sets that's relevant to all the physiology. So I might use 1,000 or 2,000 literature sources to create a model, and they're all constraining all the parameters that Michelle mentioned. It's hard to get the parameters. So that may be one way out as a more physiologically based larger model. But I wanted to ask Julien something, because Julien, you've been trying to chat.
I want to ask you a question too, but I'll let you go first.
Okay, well, the question I have for you is, you've been spot on. I mean, it really struck close to my heart when you said you're talking about predictability of models, and how do you get that in what you're doing?
So I mean, that's the most difficult part, right? And it's about trying to understand the complexity and trying to find what works in that complexity. So basically, one way to look at it is to create a network out of clinical data, and then the only way to get a sense, or at least one of the ways to get a sense on what's powerful or not, is just try to systematically trying to inactivate some genes or proteins that are of interest in your network and see what's the global output, basically. So that's experimentally one way of doing it. But everything we're going to do, we have to acknowledge the fact that it was said before, we are reducing things. Right? We just have to. We're just trying to reduce it a little bit less than typical approaches. So that's one way of doing it. Now, if you want to use like data that are already available in the clinic, for example, big data available to do some genomics, proteomics, whatever it is, one way to start to look at this is to compare a situation of a patient that have been with a disease non-treated versus treated. You start to get a sense of if you look at the data, where your biology has went, and it start to get a sense of that. But that's one way of looking at it. Now, the question I had for you was kind of when you do this modeling, do you in any way, or maybe not, attribute some value, functional value, to some parameters over other parameters? Going back to the notion of functional dominance, when you see, for example, just on paper, on publications, that whatever you're looking at, there's like 5 signaling pathways, right? Do you just like put them, all of them at the same level in your model, or it's some that actually you can prioritize?
That's a fantastic question. What we have, you're going through 1,000 literature sources. You're trying to make sense of a physiological system of disease. What we have developed in my company is a knowledge management engine where we're taking the individual data sources, evaluating them, highlighting key things, creating what we call assertions. An assertion would be something that has to be represented in the model. And then we're giving a strength of evidence score to that. And that's incorporated in a highly visible or highly accessible database, essentially, a platform that makes it easy to see these things. And then we would take those assertions and incorporate them in a master document that documents the model. And that we run by a client, key opinion leaders, specialists in the area, and they go in and they say, no, this is wrong, or yeah, you got it right here. And so now we have we get a much better idea of what's going on, and we know where the issues are. Now, once you've done the model, you can do a sensitivity analysis. So we do a local and global sensitivity analysis. We find out what models affect the outcome of what parameters affect the outcome of the model most. Now we know what the parameters that the model is most sensitive to, and we say, how certain are we of those parameters? If we're uncertain and the parameter is highly effective in changing the model output, we've got a problem. We've got a data gap. We've got a knowledge gap, and we've identified the next experiment. If everything that's the model responds to is we've got a pretty strong idea of what the parameter is, then we feel a little bit more comfortable about the predictive component of our model, as you were emphasizing the importance of the predictive power of the model. So I really appreciated that. Does that answer your question?
Yeah, it does. Thank you. Yeah, and unfortunately for us, there's going to be no quick answers to any of these. That's the nature of drug development. It's going to have to go in the clinic and go through the process of testing whatever you're doing. So
no, it's fun. It's fun. It's more fun for us.
It is fun, but it's it takes time.
Klim, one thing that you do is bring together people from different backgrounds, right? Where's an area that could be tapped for expertise that you'd really like to see more connection between that area and drug discovery? Like you mentioned, analyzing satellite imagery.
Yeah, I think that was, more of that is what I would love, right? Because again, the reality, at least from where I stand, the reality of it is, it's things like clustering and data reduction and denoising. They're sort of a very fundamental problems. And all you change is you change what your data feed in, what's the data feed in, and what's really that you feed in. And then what's the real expert knowledge that comes in is, you need a highly trained, say, cell biologist, for example, or a structural biologist to tell you this is high quality data, this you should interpret, this you should not misinterpret, etc. So that's where that expertise comes in. Once you have that person, what you really want is you want someone who maybe has no idea about biology, but is really good at, machine learning and data reduction and all this other sort of more abstract algorithmic things to work together with this person. And so, a lot of the success we had was really people coming from big tech companies, frankly, where, they use neural networks to, classify YouTube videos and things like this, and then using similar type algorithms to classify cells, classify cryo-electron microscopy particles, etc. And I would really, I really think that more of that is really what we need right now. Again, where I feel like there is certainly space for more fundamental mathematical modeling, which can then be coded, which can then be applied, but it's unclear, that's a longer term thing. I think even if biologists currently, or experimental biologists, start capturing the cutting edge of computer science today, that would be such a huge step forward. I would argue, from at least what I see around.
And it's sorry, Michelle.
Oh no, I don't want to interrupt. I wanted to ask Klim a question, and Carolina says there are a couple of questions from the audience too.
Okay, yeah. Right. how about we take some audience questions?
Sure. I can talk to Klim later.
Okay, pop up to answer. Okay, yes, I'm supposed to. Oh, there we go. It's popped up. Can you guys see the question?
This is basically what we were talking about before, huh? Creating new, more data.
Okay, anybody got an answer?
I have in the field that we're doing, there's an active area of research is led by again Nima Guy. You can look him up at Stanford, and it's really the beginning of trying to develop tools to use the breadth and the large the large nature of flow cytometry data to generate more flow cytometry data that are looking like real patient flow cytometry data. So a couple examples where we study the immune response to trauma and traumatic injury. So we've generated now real flow cytometry data from hundreds of patients, and we're trying to develop techniques. In this case, using neural network algorithm to generate in silico data that look as.
So Brice has frozen up temporarily, but I guess I understood the question to be asking whether you've got real data. Is there a way to generate realistic synthetic data?
That's a good qualification. The realistic part is
yeah.
We just a quick point in QSP, what we do is we create virtual patients, which are vectors of parameters, each of which represents a specific patient or patient phenotype, and you can do a little bit of what Brice was talking about. You can use a lot of different techniques to generate different possible parameter sets using sampling from distributions and whatnot. But then you've got to test them to make sure that they're physiologically real. It's what's done. I'm not sure that it doesn't give us data sets that are too big. In other words, I'm not sure that it's not giving us some patients that can't ever be real, but I think we get some good. I think we get some good information from the virtual patient sets that we do generate.
Yeah, certainly wasn't my impression that we were short of data, but rather it was what to do with the data. But I guess it depends on the field. Carolina, do we have a second audience question? Nevin.
Yeah.
Anyone want to respond? Michelle?
I was very fascinated by the way Klim was talking about it, really, really different disciplines, because the things that, Nevin and I do, and the drug discovery people are already very interdisciplinary, transdisciplinary, team sport, as we say. So I guess it's just finding that the thing that makes all scientists and engineers the same is that they're all curious about stuff. So if you just spark their curiosity with a hard problem, I think it's very easy to get people excited. And I'm fascinated by this idea of the hackathon, that people just come in to just try to solve a problem. It's pretty extraordinary. I don't know.
But it's
Klim,
like diving. Sorry, Klim, go ahead.
Yeah, I think there's absolutely curiosity is a major driver. I was myself, deeply surprised that, we would have this thing for 48 hours and we would have people just sort of come in and code for 48 hours trying to, classify, cells into this groups versus this, or segment nuclei or all this type of stuff, just because they're into it. I think, so it's curiosity, it's a good community. I think it all in the end comes from, there has to be, what I think helped a lot for the hackathon is trying to have some sort of social part of the event where it's not just the scientific part. But then the science needs, brings people together, and also, having impact is very, I think, important. Having medically relevant questions which are being asked, where people can really feel good about what they're doing, where they can, they can tell their, their grandparents that, I spent 48 hours working on this, and this was because it might help that. And I think that also goes quite a long way. But yeah, I think we should do more of these events, especially, now with Zoom, the whole world is connected. And so, or with all the telecommunications, I guess. And so realistically, we've been doing them very locally, but now it can be done much more globally.
And actually, I had the exact same experience. I think that the enthusiasm for the other side has been driving a lot of interest. So same thing, passion for what the other guy is doing and for the bigger goals. Same experience for me.
Do we have any more audience questions? It's, one of the challenges would seem like a welcome challenge is just different cultures, right? So in the hacker community, it's just sort of, this is what you do. You get together for 2 or 3 days straight to solve a problem. And in biology, in my memory, the way of thinking is different, because you're working with these systems that take days and weeks to change, and your rhythm of working is, you're in the lab a lot, but you're doing these repetitive procedures rather than this intense, focused problem solving. And I'm sure there's probably more curiosity on the biological side to how to bring that intensity. And then there's also just the fact that you're stepping into a field that you don't actually really know very much about. So how do you encourage people to put themselves out there and reveal their ignorance and willingness to learn something they don't know about?
Yeah.
Which I guess science is about that, though.
That's true. I'll put in a plug too, since Nevin, he's the director of QBI, and the QBI does these really cool symposia where we connect with another university, and we have the symposia where we talk about a particular topic, both sides present on that topic, and then there's opportunity for collaboration, and then even throwing some money in. So all these things, you just need to bring people together and then prime the pump a little bit. But how to just come in contact with people who do really, really different things. Yeah, that's the big challenge.
Well, this panel was pretty cool. I got to say, I very much appreciate being together with, and it's interesting too. Everybody's observing some of the same problems, but from completely different angles, and it was really a revelation to hear some of the, every single person was talking about a big data space and trying to figure out how to narrow it down, and just different ways. I appreciate being part of this, by the way, guys. So
great.
Say that now.
Does anybody have any closing thoughts? It looks like we're close to the top of the hour.
No, it's great. Well, I learned a lot.
Yeah. Well, thanks for everybody. I appreciate having this opportunity. I have learned an awful lot. So thank you to everybody. Thank you to the audience and for those questions. And I guess we're going to close off the discussion here. And of course, you feel free to get in touch with anybody if you'd like to connect, especially among the panelists. So have a great rest of your day, wherever you are.
You too. Nice job, Kaspar. Very, very, very nicely, light touch. Very nice.
You're welcome. It was a pleasure.
Thanks, everybody.
Thanks, everyone.
Bye-bye.
Bye-bye.